In this talk, we're going to learn about the early development in tunicates. Tunicates are marine invertebrate animals that belong to the phylum Chordata. To look at the life cycle of tunicates, let us first start with the fertilization of the egg with the sperm. The resulting zygote undergoes developmental processes to form a larva. The larva has a notochord and a dorsal nerve cord that were formed during early development, and this is why tunicates come under the phylum of Chordata. The larva will then attach to a substrate and undergo metamorphosis to form an adult. During this process, once the attachment to the surface happens, the organism loses majority of its nervous system. Tunicates are hermaphrodites because they have both the male and the female reproductive structures. Thus, most tunicates have both the ovary and the testes. Even though they are hermaphrodites and have ovary and testes, many tunicates do not show self-fertilization. In fact, they have evolved to have gamete incompatibility, and this does not allow self-fertilization to occur. Depending on the species of the tunicate, we can either have the egg and the sperm released in the water and thus fertilization becomes external, or we could have the sperm released in the water and these sperm then swim to the eggs that are present within another tunicate. In this situation, the fertilization is internal. The oocyte of the tunicate, as shown in the figure below in the black box, has a yellow ring-like structure called as the yellow cortical cytoplasm, and on the inner side it has a clear cytoplasm that is shown in gray, which has the yolky material. Now when fertilization occurs, the sperm enters through the vaginal pole, which is the lower part of the egg, and after fertilization occurs, the clear as well as the yellow cortical cytoplasm, they move downwards or vegetally, as shown in the figure in the black box. Now the sperm pronucleus will start moving upwards or towards the animal pole so that it can meet the female pronucleus, and in this process it drags the clear cytoplasm as well as yellow lipid-like structures that are present in that yellow cytoplasm, and this results in the formation of a yellow crescent as shown in the figure in the black box. Thus, there is a portion of the zygote that has a yellow tinge, and this portion of the zygote has been shown to form the tail muscles, which we've learned about when we learned about autonomous specification in Chapter 2. Tunicates show bilateral holoblastic cleavage, and what we mean by that is the first cleavage plane is able to establish an axis of symmetry. So when we look at the figure that's there in the black box, there is an axis of symmetry that has formed that is able to split the embryo into the right and left sides. This is more evident in the figure that's shown in panel C, where we're able to see that axis of symmetry and we're able to see the left and the right side as mirror images as one another. As the organism further undergoes the process of cleavage, it's very clear that the right and the left sides have been established during the process of cleavage. Thus, tunicates are able to show bilateral holoblastic cleavage. In the case of tunicates, the zygote shows the presence of a yellow coloration or a yellow crescent. The presence of this yellow coloration can allow us to map the fates of the cells that will form in that region. This is exactly what the embryologist Conklin did when he noticed that cells with the yellow color actually develop into muscle cells. Conklin was able to map all the early cells during tunicate development and was able to determine their fate, and thus the parts of the embryo can be predicted in the zygote itself. Thus, tunicates are known to show autonomous specification. As part of his experiment, Conklin cultured the individual cells from the 8-cell embryo and determined the fate of each of the cells in this 8-cell embryo. For example, when he cultured the B4.1 cells, then he found that those cells resulted in the form of muscle tissues. Since they formed the muscle tissues and not the entire embryo, this showed that the cell B4.1 underwent autonomous specification. Furthermore, the removal of the B4.1 cells resulted in the absence of tail muscles in the developing embryo. This is true for a lot of other cells also, where they already know their fate very early on in development. Thus, tunicates are a great model to study autonomous specification. Following cleavage, gastrulation occurs, and during the early stages of gastrulation, a notochord is formed. As development progresses, the dorsal root ganglion also develops, and the notochord and dorsal root ganglion development are characteristics of all organisms that belong to the phylum Chordata. As development progresses in the case of tunicates, organs form and we end up with a larvae. The larvae, as we are shown in the figure, has different structures which includes the notochord as well as the nerve cord, which help the larvae swim around in the ocean or in their environment and find an appropriate substrate. Once the larvae find an appropriate substrate, they're able to anchor to that substrate. Following that, metamorphosis occurs. Metamorphosis results in the formation of the adult tunicate. During the process of metamorphosis, most of the nervous system features undergo degeneration because they are no longer used. With this, we come to the end of our talk, where we learned about the life cycle of tunicates as well as fertilization and cleavage in tunicates. We also learned about the formation of the yellow crescent in tunicates and how tunicates are used to study autonomous specification.